Assalamualaikum and good morning to Madam Munirah. My name is Adil bin Muhammad Anwar. My metric number is 230F1016. So now I'm going to do a virtual bacteria identification lab. So the objective is we will find kind of patient samples are used for the purpose of identifying possible pathogens. And the other objective is what does PCR do and how does it work and why is it useful, and how do you separate the desired DNA from all others, and how does an automatic DNA sequencer work, and lastly, why is it possible to use a DNA sequence to identify bacteria. So now we will begin the lab. So we will start over the lab virtual. So firstly, we will prepare a sample. To begin, we will put a pair of rubber gloves by clicking the glove box in the open drawer. And then we will obtain a sample from the culture dish and we will click the wire loop to continue. So we will pick one of the bacterial colonies. So now we will click the microcentrifuge tube to transfer the bacterial colony from the wire loop into the microcentrifuge tube. And now we will add digestive enzyme to our sample by clicking on the yellow micropipette to continue. And then the microcentrifuge tube must sit for several hours. And then after several hours of enzymatic digestion, our sample is ready by clicking the microcentrifuge tube to hit inactive the digestive enzyme. And then the digestive enzymes are now denatured and inactivate by clicking the microcentrifuge to continue. After centrifuge is complete, we will upload the centrifuge. Unwanted debris have now been spun down. Then A1 is in the supernatant and click the microcentrifuge tube to continue. Now we are transferring the supernatant to the PCR tube. So the sample preparation is complete. Click the orange tray to continue into part 2 for PCR amplification. And now we will add the PCR master mix solution to all tubes. Click the red cap master mix bottle to continue. And then we need to set up the positive control reaction. So now we are adding control DNA to the positive control PCR reaction tubes. And for the negative control reaction, we will add water. So we will click the PCR machine to open the loading compartment and load the PCR tubes. So the PCR tubes have successfully been loaded into the PCR machine. and then we will running PCR amplification. So the PCR amplification is complete. We'll click the orange tray to continue to the PCR product purification process. Okay, now for part 3, that is for purify PCR product. First, we will set up the micro concentrator column by click the yellow cap buffer bottle. Continue. So now we're adding buffer solution to the column. So we'll click the orange tree to continue. So the tubes containing the PCR sample of positive control and negative control will be put on ice. So DNA is now trapped in the column. Now we need to loosen it into a new tube. So we'll adding buffer solution to the inverted column. So the first collection tube is no longer needed, and we'll discard the first collection tube. So now we will centrifuge the assembly to collect the DNA into the collection tube. So loading the column into the centrifuge. Centrifuge it for 2 minutes. So now DNA has now been collected in this collection tube. So the column is no longer needed and will discard. The PCR product has been purified, and we'll go to the next part for DNA sequencing. So for prepare for sequencing, first we will dilute the PCR product to increase the volume. We will click the blue cap distilled water bottle to continue. So the green and blue strip tubes already contain PCR reaction mixtures in them. So we are adding purified PCR product to the strip tubes. So the strip tubes are ready for PCR amplification. So loading the strip tubes. So we're running PCR amplification. So the sequencing preparation is complete. I will continue to part 5 for sequencing preparation. So now we will DNA sequencing, and first we will load the automatic sequencer. So we will click the integrate door above the orange tray to view the DNA sequencing animation. Okay, now is sequence. So we have completed the DNA sequencing section. So the sixth part is DNA sequence analysis. So we click complete screen. We identify the bacteria. So the correct bacteria is C. Bartonella henselae.